Welcome to Biotanemy. Necrosis.
Necrosis, or regulated necrosis, is a programmed form of necrotic cell death, which leads to cell swelling, plasma membrane rupture, and the release of DAMPs, or damage-associated molecular patterns. Molecular signaling pathways actively control the process. Different types of necrosis are 1. Necroptosis 2. Pyroptosis 3. Ferroptosis 1. Necroptosis. Necroptosis is a form of programmed necrosis that occurs when apoptosis is inhibited. It is triggered by TNF-α, or tumor necrosis factor alpha, binding to TNF-R1, or the TNF receptor, which leads to the recruitment of adaptor proteins TRAD, or TNF receptor-associated death domain, TNF receptor-associated factors, cellular inhibitors of apoptosis proteins, or CIP1, and RIPK1, or receptor-interacting protein kinase 1, to form Complex 1. Complex 1 plays a pro-survival role through NF-kappa B. Complex 2 forms on ubiquitination of RIPK1. Upon ubiquitination, RIPK1 will dissociate from TNF-R1 and recruits FADD, caspase 8, and RIPK3. Recruited RIPK3 will phosphorylate MLKL, or mixed lineage kinase domain-like protein. Phosphorylated MLKL oligomerizes and translocates to the plasma membrane. MLKL disrupts membrane integrity by punching holes in the plasma membrane. The cell undergoes necroptosis, releasing DAMPs, or damage-associated molecular patterns, and triggering inflammation. 2. Pyroptosis. Pyroptosis is a highly inflammatory type of programmed cell death triggered by intracellular infections, toxins, or danger signals. It is primarily mediated by inflammasomes and executed by gasdermin proteins, leading to cell lysis and the release of pro-inflammatory cytokines. Pyroptosis begins when pattern recognition receptors, or PRRs, detect pathogen-associated molecular patterns, or PAMPs, or damage-associated molecular patterns, or DAMPs. These signals activate inflammasomes, such as NLRP3, or NOD-like receptor family, pyrin domain-containing 3, AIM2, or absent in melanoma 2, and LRC4, or NOD-like receptor family, CAR domain-containing 4. Activated inflammasomes recruit caspase 1, leading to its autocatalytic cleavage into active caspase 1. Caspase 1 processes IL, 1 beta, and IL, 18 into their active forms IL, 1 beta, and IL, 18, which are potent inflammatory cytokines. Caspase 1 cleaves gasdermin D, or GSDMD, releasing its N-terminal domain, which inserts into the plasma membrane and forms pores. These pores allow water influx, ion leakage, and cell swelling, leading to plasma membrane rupture. IL, 1 beta, and IL, 18 exit through GSDMD pores, triggering an inflammatory immune response. DAMPs like ADP, HMGB1, and mitochondrial DNA are also released, further stimulating inflammation. 3. Non-canonical pyroptosis. Non-canonical pyroptosis occurs when caspase 4/5 in humans directly recognizes cytosolic LPS from gram-negative bacteria. These caspases are activated independently of inflammasomes upon binding with LPS. Active caspase 11/4/5 cleaves gasdermin D, GSDMD, at its linker region. The GSDMD fragment translocates to the plasma membrane and forms pores, leading to cell swelling and rupture, the release of IL, 1 beta, IL, 18, and other inflammatory signals. 4. Ferroptosis, or iron-dependent cell death. Ferroptosis is a unique, iron-dependent form of regulated cell death characterized by lipid peroxidation and oxidative damage. Ferroptosis is driven by iron overload, ROS accumulation, and lipid peroxidation. Rapidly dividing cells, like cancer cells or immune cells during inflammation, have a higher demand for iron, needed for DNA synthesis, respiration, and metabolism. Most cellular iron enters as ferric iron bound to a plasma protein called transferrin. The transferrin-ferric iron complex binds to transferrin receptor 1 on the cell membrane. This complex is then endocytosed into the cell. The divalent metal transporter 1, or DMT1, transports ferric iron from the endosome into the labile iron pool, or lip, in the cytosol. Excess ferrous iron reacts with hydrogen peroxide via the Fenton reaction, generating reactive oxygen species. ROS causes oxidative stress and lipid peroxidation in cell membranes, forming lipid peroxides, or LOH. LOH breaks down into reactive aldehydes, like 4-HNE and MDA, which disrupt membrane integrity, damage proteins, and DNA. Excessive lipid peroxidation leads to membrane rupture, releasing DAMPs.
That's all for today's video. Thank you for listening. Hope all of you understand the concept, and for more queries and proposal, please comment down. See you all in a new topic.